biotechnology.
Phytochemicals from Oxymum gratissimum leaves, and we study molecular docking between these plant manner.
ये हमने post like which is antibiotics.
respected dignitaries on the dais, respected invited guests, respected faculty members, and dear participants, a very good morning to one and all. I, Dr. Harshita Shukla, Head Department of Biotechnology, Shri Guru Tegh Bahadur Khalsa College, Jabalpur, and Organizing Secretary of the first International Conference on Nature and Natural Sciences, extend a very warm welcome to all of you. Today, we are all gathered here for the second day oral online presentation session. The chairperson of today's session will be respected Dr. Anju Pathak Madam, IQAC Coordinator, Shri Guru Tegh Bahadur Khalsa College, Jabalpur, and co-chairperson will be Dr. S.K. Dubey Sir, Shri Guru Tegh Bahadur Khalsa College, Jabalpur. The instructions for the participants for today is that all the participants will be given a time of 5 minutes for their presentation. Kindly concentrate on the title of your research work and the findings of your research work because we have a specific time frame for this session, which will be followed by the valedictory session. So, beginning with today's program, I will call out the first participant of today, Miss Swapna Mishra, Assistant Professor, Government Jwala Yadav Naveen Girls College, Jangir, Chhattisgarh.
Good
Madam
morning.
Swapna Mishra. Okay.
Good
Ma'am,
morning to all.
please share your PPT.
Yeah. On first international conference, Nature and Natural Science, myself, Swapna Mishra, Assistant Professor of Zoology, Government Jajwal Dev Naveen Girls College, Janjgir. Topic of my presentation is agrochemical toxicity and fish fauna. स्क्रीन शेयर हो रहा है मैम?
Yes, ma'am.
जी, थैंक यू.
Ma'am, please directly tell
जी
your
मैम.
title and
जी
then
मैम.
the conclusion of your study. Please skip
जी.
the introduction and methodology part.
जी. एग्रोकेमिकल्स, ये हमारा जो प्रेजेंटेशन है वो प्री रिसर्च पर डिपेंड कर रहा है. पूरा उसी पर हमने बनाया है और ये हमारा मेन ऑब्जेक्टिव यही है, कलेक्ट इन्फॉर्मेशन अबाउट हार्मफुल इफेक्ट ऑफ एग्रोकेमिकल ऑन फिश फौना. मोर्टेलिटी पर देख रहे हैं, बायोकेमिकल पैरामीटर, बिहेवियरल रिस्पॉन्स, हिस्टोपैथोलॉजिकल पैरामीटर पर. सबसे पहले मोर्टेलिटी, ये पेस्टिसाइड फिश फौना पर कैसे इफेक्ट करता है, कौन कौन से पॉइंट पर इफेक्ट करता है. टॉक्सिसिटी, LC50 वैल्यू देखी जाती है. लीथल कंसंट्रेशन इज द अमाउंट ऑफ सब्सटेंस रिक्वायर्ड टू किल 50% ऑफ टेस्ट एनिमल ड्यूरिंग प्री डिटर्मिनेंट ऑब्जर्वेशन पीरियड. चूंकि हमारा ये प्री रिसर्च वर्क पर बेस्ड है तो इसने हमने प्री रिसर्च वर्क पर लिया है LC50 वैल्यू का. साइपरमेथ्रिन, मेलेथियोन, एंडोसल्फान, डाइमिथियोनिन टेस्ट ऑर्गेनिज्म पर 96 आवर ड्यूरेशन पर पेस्टिसाइड का इफेक्ट देखा गया तो LC50 वैल्यू अलग अलग आई है. बिहेवियरल रिस्पॉन्स यदि देखा जाए तो बिहेवियर मेन टॉक्सिकोलॉजी का मेन बिहेवियर होता है. बिहेवियर देखने के लिए एबनॉर्मल बिहेवियर, न्यूरोटॉक्सिक इफेक्ट इस पर शो करता है और जो भी न्यूरोट्रांसमीटर है, behavior main है very promising tool in ecotoxicology. Abnormal behavior show by fish may be due to abnormal level of neurotransmitter. जब भी पेस्टिसाइड या एग्रोकेमिकल किसी भी फिश पर आता है तो सबसे पहले नर्व सेल बॉडी एक्सॉन माइलिन शीट को डिस्ट्रॉय करता है जिससे न्यूरोट्रांसमीटर की सिंथेसिस और रिलीज में फर्क पड़ता है. ये हमने प्री रिसर्च वर्क से लिया है कि कौन कौन से पेस्टिसाइड पर बिहेवियरल चेंज कौन कौन सा आया. तो मेनली बिहेवियरल चेंज आता है, स्विमिंग एक्टिविटी डिक्रीज हो जाती है, इरेक्टिक स्विमिंग आती है, सरफेसिंग आता है, पूरे बॉडी पर म्यूकस डिपोजिशन बहुत ज्यादा होता है, ह्यूमेरिया होता है. तो इस प्रकार इम्बैलेंस स्विमिंग. फिश में सबसे पहले बिहेवियरल टॉक्सिकोलॉजी देखी जाती है. हिस्टोपैथोलॉजिकल अल्टरनेशन, वेरी यूजफुल टूल फॉर टॉक्सिकोलॉजिकल स्टडी. Monitoring tissue alteration, pathogenic change in fish exposed to different concentration of एग्रोकेमिकल। इसमें लीवर बहुत ही इंपॉर्टेंट रोल प्ले करता है क्योंकि डिटॉक्सिफिकेशन में काम आता है, इसलिए स्टडी में लीवर को मेनली लिया गया है। जब लीवर की स्टडी देखी जाती है हिस्टोपैथोलॉजिकल चेंज, तो मेनली इसके हिपेटोसाइट्स के शेप और साइज में चेंज आता है, रैप्चर हो जाती है, हिमोरिया हो जाता है, मोर नंबर ऑफ वैक्यूल्स का फॉर्मेशन होता है, इनलार्जमेंट, लीवर सेल्स बड़ी हो
Madam,
जाती है।
please come to the conclusion, ma'am. What are the findings of your study? What is the
बायोकेमिकल
conclusion?
बायोकेमिकल पैरामीटर में प्रोटीन। कंक्लूजन, इस
Yes,
प्रेजेंटेशन
ma'am.
मेनली बेस्ड है
Please
अर्लियर
highlight the conclusion.
tried to shed light on toxic effect of agrochemical on fish farm. एग्रोकेमिकल पूरी तरह से डिपेंड हिस्टोपैथोलॉजिकल, बायोकेमिकल और बिहेवियरल चेंज करता है। इस तो गवर्नमेंट को स्ट्रिक्ट रूल्स डालने चाहिए एग्रोकेमिकल के यूज के लिए। हार्मफुल पेस्टिसाइड मस्ट बी स्ट्रिक्टली enforced by imposing jail and massive fine। जो हार्मफुल केमिकल हैं उनके यूज के लिए इनको बहुत ध्यान रखना चाहिए कि स्ट्रिक्ट रूल इनको फॉलो करने चाहिए। ये रेफरेंसेस है जो हमने अपने इसके लिए बनाया हुआ है। थैंक्स फॉर योर अटेंशन।
Thank you, ma'am. Next participant, Miss Ananya Gupta, Gautam Buddha University, Greater Noida.
Good morning, ma'am.
Please share your PPT, Ananya.
Yes, ma'am.
Ananya, please share your PPT.
Yes, ma'am. I'm sharing. There's some issue with me. Ma'am, is this visible?
Yes, it is visible. Okay. Please tell your title and conclusion of your study.
Okay, ma'am. So good morning to all the chairpersons and rapporteurs of First International Conference of Natural Sciences. The topic for my presentation is groundwater quality assessment using Water Quality Index of Manipur, India. So the groundwater is a valuable and widely distributed resource of the planet. It is widely used all over the realm, and every citizenry relies on it, as we know. But due to depletion, severe depletion, and due to depletion, there have been a loss of sorry, my bad. So groundwater supply and quality has been impacted due to urbanization, particularly in developing countries like India, due to overexploitation and inappropriate waste disposal, mainly found in metropolitan city. Hence, we conducted a studies in Manipur state where we took 11 samples from different states of different places, sites, where we conducted studies on groundwater quality of Manipur state based on the results and presented the result of physiochemical and hydrogeo and bacteriological analysis. This research is significant since Manipur is a mercantile with fast urbanization and development, and this region is densely populated and has impacted on groundwater due to overexploitation. Hence, I can present my findings. This was my methodology. We collected the sample, and the results were we found that the TDS of the water was in the range of 56.60 to 623, which was within the some of the samples were within the allowable limit, but maximum TDS value was found in R1 and R9 samples. The salinity, TDS, and electrical conductivity all are interconnected. Hence, it was presented that minimum and maximum value of electrical conductivity obtained in samples were R22 and R19. So, upon coming on my conclusion, we also calculated for cation concentration and analysis of anion. You can see the graphs presented, pictorial representation of all the analysis. Here, we represented the data in the form of Hill-Piper diagram and Duroff's plot with help of Gibbs diagram. flow of anion and the interaction of anion with the rocks as well in the which is present in the water, and the results were supported with Chada's plot as well. The the result was important. It was important to conduct the study because water is very important for irrigation and drinking purposes. The permissible limit which was a permissible limit which was you what do you say? The permissible limit which is required is very important for irrigation purposes since it impacts the soil quality and impacts the crop cultivation. In the present study, the water quality index is found to be 196, and 31.8% of the sample are found in the good range of consumption, except some of which show poor quality. The results of Hill Piper plot and Grove's plot supported by Chadda's plot suggested alkaline earth metals dominant dominant than alkaline metal, therefore indicating bulk samples having temporary hardness. Gibbs diagrams and relative statistical analysis.
Ananya, your time is over.
Ma'am, just a minute. Can I get another minute?
Okay, we are just giving you last minute. Please conclude your work.
Yes, ma'am. Okay. The and relative statistical analysis of diagrams show that rock weathering and ion exchange have played the major role in hydrogeochemistry. Based on the result of collected analysis, the periodic measurement of various water quality parameters may assist and enhance the water treatment process. It must be realized that water testing is a vital approach for ensuring availability of contamination-free drinking water while also encouraging citizens to become more aware of sanitation and hygienic conditions. Hence, the above study makes it a valuable database for future references. So here are my references. Thank you.
Okay, thank you, Ananya. Next participant, Miss Ankita Rani, St. Joseph University. Miss Ankita Rani, are you there? Okay, we will move to the next participant, Mr. Kartavya Mathur, Gautam Buddha University. Mr. Kartavya Mathur.
Yes, ma'am. Sharing my slide. Ma'am, is my slide visible?
Yes.
Good morning, everyone. Today, myself, Kartavya Mathur, master's student from Gautam Buddha University, is here to present my research work titled In Silico Analysis of Algal Metabolic Models for Enhanced Biofuel Production in Chlorella vulgaris. A little background of the project. We all know that fossil fuels are in a rapid declining stage, and they're also a great contributor to global warming. Due to this reason, scientists have recently started looking for better renewable and sustainable energy sources, which can be extracted from various microorganisms such as algae, bacteria, fungi, due to their lower greenhouse gas emission tendency, greater renewability, and high energy security. Algal are algal species are prospects for biofuel production due to their higher productivity, versatile feedstock, and greater lipid content, and very significantly studied microorganisms. Here we intend to target Chlorella vulgaris because it has great tendency to produce bioethanol, has comparatively higher nutrition value, has ability to fix carbon dioxide, and has tolerance to environmental conditions. Here we present a systems biology approach to simulate the model for chlorella for increasing or enhancing the biofuel production.
Kartavya, please highlight your conclusions,
Yes, ma'am.
findings and conclusion of your work.
Yes, ma'am. So this was our methodology where we first simulated, first we collected the models, genome scale metabolic models from from a recently published paper. These are the statistics of the model. Here we targeted pyruvate metabolic pathway of Tora verbalis variabilis, which is a reference genome for Florida species. We simulated our model on various growth substrates such as glucose, succinate, ethanol, etc. And we identified that glucose, ribose, and L-glutamine showed an increase in biomass productivity. Next we tried to correlate glucose uptake with growth rate and oxygen uptake with the growth rate as well as bioethanol production. Here we can see that glucose is directly correlating with the increased growth rate as maximum growth rate at 100 millimole per dry weight per hour reaches up to 3.75, and for similarly there is a steady increase in growth with increase in oxygen concentration. However, this is not the case with bioethanol production as increasing oxygen uptake shows a negative effect on ethanol production. We substantially we used single gene knockout analysis in silico removed where we generated a matrix for or deletion of each gene and plotted its GR ratio, which is the ratio between flux growth rate at gene knockout stage and growth rate at wild type organ.
Kartavya, please come to the conclusion slide
Okay, ma'am.
and tell your conclusions.
So our key findings from the study were that using glucose, ribose, and glutamine as a substrate, we can substantially increase the biomass productivity of a cluster of chlorella. Further, we correlated. We can say that glucose uptake can be correlated with ethanol stress. Basically, oxygen stress can be correlated with ethanol stress. Gene essentiality analysis showed that biomass precursors included several glycerides, ATP, ADP, rhodopsins, and bioethanol production was majorly linked to amino acid transporters, acetyl CoA transporters, acid exchanges, and several key biomass precursors. Flux variability analysis of 6 genes involved in pyruvate metabolites suggested that these reaction can substantially removing genes involved in such reaction can substantially increase our bioethanol flux. The future aspects of the study we tend to study more combinatorial analysis of substrates to find best substrate for ethanol production, incorporating butanol pathway as there are key enzymes which we have identified in the pathways directly that which are linked to butanol production, and we then propose to experimenting this uh, genetically engineered or genetically updated species in lab.
Okay,
These were the
Kartavya. Time is over, Kartavya.
Yes, ma'am. Thank you.
Okay, thank you. Next participant, Dr. Niranjan Kumar, Assistant Professor, Kamdhenu University, Gujarat. Dr. Niranjan Kumar,
Yes, ma'am.
please share your PPT.
Okay, ma'am. Good morning, all of you. I am going to present the research topic detection of nucleic acid of the Anaplasma marginale in the tick vector.
Your PPT is not visible. I think your computer screen is visible, but your PPT is still not visible.
Now, ma'am, visible?
No, it's not visible. ओपन नहीं है आपका पीपीटी, प्लीज ओपन इट.
Now, ma'am.
No, it is not visible. Okay, you can just tell your title and conclusion orally.
I am going to present the research topic detection of nucleic acid of Anaplasma marginale in the tick vector. And objective of this our research is depending upon the background information. Objective of the research is detection of Anaplasma marginale in the Ripicipilus bupilus microplus tick of the cattle using species specific PCR targeting MSP5 gene. And finding of our research work in the Giemsa stain, the thin blood smear of the Anaplasma marginale appear as a blue color and the basophilic inclusion body near the margin of the erythrocyte. Here, ma'am, in our research, resolve the PCR product under the UV light and 1.5 agar gel following the staining the ethidium bromide. And we using also a eco R1 enzyme cleavage the 576 base pair of the MSP gene of the Anaplasma marginale at the specific site into two fragments, and we visualize agar gel and the digestion additional confirmation of the species specific of the PCR for the Anaplasma marginale. We can also using the vector DNA isolation, and we confirm this in our research around the 52 group of the Ripicipilus microplus bupilus tick collected from the our area and examined for the PCR after the following the thin smear examination, and out of them 52 group 48, 44, 46 and 18 were observed in carry the Anaplasma organism in their whole body, anterior half, posterior half and egg in the PCR targeting the 16 srRNA genes. Whole body of the 42 out of 48 group of the tick with Anaplasma organism detect MSP5 gene fragment of the Anaplasma marginale. Multiple sequencing alignment of the two sequence of the MSP5 gene of the 470 base pair long Anaplasma marginale using the omega program and alignment showing the similarity between the sequencing. The NSBI blast analysis of the current two sequence of the MSP gene of the Anaplasma marginale show the 100% identify with the published nucleated sequence. A mass scalar total score of 771 and E value of this 0. This indicate the highly significant match. This confirm the accuracy and reliability of the two sequence. And also we prepare a phylogram and analysis is performed using the MSP5 gene of the Anaplasma marginale. The analysis involve total 1 to 7 are alignment and 8 and 9 are different. So this is for the Anaplasma non-pathogenic organism. And lastly we conclude topic. The Ripicipilus bupilus microplus tick have the potential to transmit the Anaplasma marginale, which is significant threat to the health of the cattle. MSP5 gene is a potential a marker to identify the Anaplasma marginale in the tick in PCR using the species specific primer with the product size of the 470 base pair and 576 base pair. NCBI blast analysis of the current sequence of the MSP5 gene of the Anaplasma marginale show the 100% homology with the published sequence. And last conclusion, PCR with the MSP5 gene primer combination prove the effective in the detecting the presence of the Anaplasma marginale in whole anterior posterior half of the body of the infected Rhipicipilus bufilus microplus, including in the egg, indicating the trans-ovarian transmission of this rickettsial agent. Thank you, ma'am.
Okay. Thank you, sir. Next participant, Miss Divya Das, the Maharaja Sayaji Rao University, Baroda.
Yes, ma'am.
Please share your PPT, Divya.
Is it visible?
Yes, it is visible.
Okay. Good morning, everyone present over here. Today, myself Divya Das from MS University Baroda is here to present my topic, that is survey of fruit waste management in some fruit-based industries of Gujarat. Fruits are indispensable part of our daily life as they are source of essential nutrients to maintain our body healthy. With the increasing population as well as shifting demographics, there has been abundant increase in fruit production all around the world. 50% of the produced fruit are converted into juice and rest into processed products. Thus, at last, industries are left over with huge amount of fruit waste generation that generally includes seed, peel, pomace, etc. However, these fruit waste are rich with various type of bioactives that can further be valorized into useful byproducts, thus leading to waste minimization. In the present study, the survey of 10 mango-based industries of Surat district of Gujarat state has been done. So why mango? Mango belonging to family Anacardiaceae hold
Please
its
tell your conclusions, Divya.
uh, yes, ma'am.
Conclusions of your study.
Yes, ma'am. So the fruit has its own importance at the state level. So major varieties have been found out my study. The telephonic survey of 22 industries of Surat district has been done with the main objective to identify actual mango varieties used by the industry, place of their procurement as well as their various management practices, waste management practices. Out of 22, 10 industries has responded with giving their information about their waste management practices as well as the mango variety they use. Coming to the result, out of 10 responded industries, 8 industries uses ripe mango varieties of Kesar, Alfonso and Totapuri mango varieties for making their various products, generally coming from the Junagadh, Valsad district as well as Totapuri variety coming from the South India. Out of 10 industry, 5 industries that uses raw mango for making pickle and amchur powder uses the varieties of Rajapuri, Kesar and wild varieties of Junagadh as well as Valsad for making their various products. Talking about the waste management practices of the industry, so majority of the industry that are left over with the seed uses the seed for making mouth freshener as well as some of them also sell that to the nursery and use this leftover seeds for making biogas production as well as the fertilizer. Coming to the peel, majority of the industries use peel for making biogas as well as fertilizer. If the industries have a small amount of peel left over, so they use it in the form of cattle feed as well as they directly discard them also. Coming to the discussion, the fruit has 35 to 60% of the weight made up of mango peel as well as seeds. Various researches have been going on this bio waste. According to recent findouts, mango peel is a good source of various vitamins, carotenoids, polyphenols having strong antioxidant qualities. Also, the seed kernels possess different types of essential amino acids, phenolics, phytosterols, etc. Thus, by characterizing and valorizing this leftover mango fruit waste into some useful byproducts can be a turning point in food, cosmetics and pharmaceutical industries worldwide with the promise of returning economic benefits as well as waste minimization, thus finally leading to the sustainable development. Furthermore, enhanced research works can be done on various varieties of mango. Thank you so much.
Okay, thank you, Divya. Next participant, Mr. Mansoori Sanobar Mahmood, the Maharaja Sayaji Rao University, Baroda. Mr. Mansoori Sanobar.
Yes, ma'am. Am I audible?
Yes, please share your PPT.
Ma'am, is the screen visible?
No, it's not visible. Now it is visible. Please open your PPT.
Ma'am, is the
Yes.
screen visible?
Yeah, it is visible. Tell your title and then conclusions of your work directly.
Hello.
Yes,
Uh,
please start.
okay. Good morning, one and all present here. Myself, Sanobar Mansuri from the Maharaja Sayajirao University of Vadodara is going to present my topic on evaluation of fungal strain tolerance and its potential to degrade pH. So we know that environment in the environment pH, that is polycyclic aromatic hydrocarbon, are among the most prevalent organic pollutant. They contaminate environment widely, necessitating the development of efficient remediation technique. It gets liberated into the environment by pesticide, fungicide, detergent, dyes, and mothball, and has been carcinogenic and mutagenic effect. BH, being extremely fat soluble, gets easily absorbed from the mammalian digestive system and quickly spread out in the variety of tissue. So they need to be eliminated from the environment. So various techniques have been developed in the year. So to eliminate these contaminants. So my work mainly focuses on using the bioremediation techniques, mainly the fungi, for degrading these pH. So these are the some previous works done in these topic. So this work mainly focuses on using fungi as a potential degraders to tolerate the contaminated soil. Firstly, the fungi were isolated using two different media, that is
Please
potato
tell the
dextrose agar
conclusion,
and
Divya.
Apecox media.
Mansuri.
Best suited media was then further used
Mansuri,
for the
please tell
subculture. Later, the
conclusions.
fungi were
Skip the
inoculated with MS
methodology.
media supplemented with 3% of petrol, both in solid as well as in broth media. So the
Mansuri
results
Sanobar.
show that both the graph depicts the growth of fungal strain on both the media, where it was observed that except for the SA5 and SA19, all the other strain were more compatible to the potato dextrose agar media than the Zapec media. After the incubation on the media supplemented with the petrol, the tolerance rate of the fungi were calculated using the formula given by the Anahid et al. That is FTR, that is fungal tolerance rate, is equal to diameter of fungal growth in the test plate divided by diameter of the fungal growth in control plate into 100, which shows that SA13 and SA14 showed the best tolerance index, that is about 90% for solid media, while SA7, SA10 and SA12 had a tolerance index above 70%. So similarly,
Mansuri,
the
please
fungi were
concentrate on
inoculated in the broth
conclusions.
media, where it showed the
Am I
SA16,
audible to you,
SA6 and
Mansuri
SA9 showed the
Sanobar?
highest tolerance in the broth media. So, from the above study, it can be concluded that the PDA media and the broth media are better for the growth of fungi and finding the tolerance index respectively, while SA13, SA14, and SA16 were identified as the possible biodegraders in the current study's primary screening test. Moreover, more research is needed to assess this fungi ability to degrade the pH, particularly the various factors involved in the efficient degradation. Furthermore, additional research is needed to understand the pH removal mechanism in order to manage the fungal resources to mitigate the effects of the pH contamination. So these are my references. Thank you.
Thank you, Mansuri. Next participant, Mohammad Asif Beg, Assistant Professor, Shri Guru Tegh Bahadur Khalsa College, Jabalpur.
Morning, ma'am.
Please share your PPT, sir.
Yes, ma'am.
Your PPT is not visible, Asif Beg.
Is it visible now?
Yes, it is visible.
Okay.
Please
Very
tell
good.
the title and conclusions of your work.
Okay, ma'am. Very good morning to respected jury members and the participants who are present here. On the basis of today's theme, my topic is the in vivo studies of antimicrobial, phytochemical, and silver nanoparticle studies from Ficus religiosa imp. These are the contents, and the conclusion of my work is the conclusion of my work is the project deal with the microbial and in vivo evolution of Ficus religiosa with respect of three broad areas of work. And even though this tree has an important significance in a traditional system of medicinal sense, ancient time, there is an increasing need to investigate various important aspects of the multipurpose tree of as to make it important beneficial of mankind. Uh, Antimicrobial activity assay with various solvent extract of in vivo leaf, twig, and aerial root shows that this tree has high activity against the five infectious organisms that were tested. Well diffusion method was found to give a better result compared to the disk diffusion method. In conclusion, it was clearly observed that all parts of Ficus religiosa include the in vitro rhizosis callus culture had good antimicrobial activity, confirming it property of a medicinally important tree species. Phytochemical analysis of the various in vivo part of the tree reveal that Ficus religiosa is a rich source of a large variety of phytochemicals. The aqueous and ethanol extract of the in vivo leaf, twig, and aerial root were tested qualitatively for the presence of phytochemical compounds such as phenolic, tannin, and flavonoid, etc. It was found that alkaloids were present in leaf, twig, and aerial root, whereas saponins were found in the aqueous extract in vivo parts and in the ethanol leaf and aerial root of extracts. Uh, silver nanoparticles that were synthesized were found to have a maximum absorbance at the range of 430 nanometer, which correspond to the surface responses peak similar to that of silver nanoparticles. SAM analysis showed the particle size of the nanoparticles between 25 to 50 nanometer. The antimicrobial activity assay showed that the nanoparticles were highly effective against the three bacterial species tested. For future scope, thus it can be concluded that all the work carried during the present study will be a landmark of future studies involving isolation and identification of the bioactive compounds. This study shows the antimicrobial properties to twin for first time along with leaves. The bioactivity molecule from the zone of inhibition could be evaluate and characterize further. Also, study can be carried out to check if the particular phytochemicals in the active in the cured form of or if it has to be purified further for increase the innate activity. Quantitative analysis of the extract should be done to check the amount of bioactive activity present in the plants. Green synthesis of silver nanoparticles can further to be optimized and synthesized can also be done from other plant parts, which can be helpful in further in drug delivery system. Okay, thank you, ma'am.
Okay.
These are the
Okay, sir.
differences.
Thank you. Next
Okay.
participant, Dr. Savita Tiwari, Assistant Professor, Shri Guru Tegh Bahadur Khalsa College, Jabalpur. Dr. Savita Tiwari, are you there? Okay, we will move to the next participant, Miss Vasundhara Singh, CMP PG College, University of Allahabad. Vasundhara Singh. Next participant, Mr. Ripu Daman, Research Scholar, Chaudhary Bansilal University, Bhiwani.
Yes, ma'am.
Please share your PPT.
Is it visible, ma'am?
No, it's not visible.
Good morning, each and all. My title is Some Sustainable Ways to Conserve Biodiversity. Uh, what is biodiversity? The variety of animals, plant, fungi, and microorganisms that all living beings that are present in our ecosystem is known as biodiversity. Biodiversity is important as it is connected to nature, and whatever is there present is there is present for their survival. How can we conserve biodiversity? Protecting and managing biodiversity in order to gather resources for sustainable development is known as biodiversity conservation. We have to conserve biodiversity because everything and each and everything we have seen in our nature is to be conserved, because there are so many disasters which are coming due to lack of our management. So we have to conserve biodiversity. There are three goals so we can conserve biodiversity: to maintain the variety of species, sustainable use of ecosystem and species, to protect vital ecological processes and systems that support life.
Ripudaman,
How?
please tell conclusions.
Okay, ma'am. Uh, by planting trees and flowers which are native origin, we can conserve biodiversity. By bees play an important role in many ways. It helps in pollination, so we have to protect bees because there are many parasites which attack on them. We have to promote local agriculture, which have to be encouraged so that we can conserve our biodiversity. Local habitat, which include many trees and plants of that area, should be conserved, which are going to be endangered nowadays. Conclusion: nature has given all the resources for fulfilling the conditions which are necessary for living. But nowadays, due to urbanization and increased use of resources for human greed, we are going toward depletion of our resources, which causes a degradation in the quantity and imbalance in nature. So there is a need arises to conserve these resources and to conserve the biodiversity. We need some sustainable methods, which I have discussed above, so that we can conserve our biodiversity and our ecosystem.
Okay. Thank you, Ripudaman.
Thank you, ma'am.
Next participant, Miss Rashmi Kesharwani, Banaras Hindu University.
Yes, ma'am.
Please
I'm
share your
audible to you?
PPT. Yes, you are audible. Share your PPT.
Yes, ma'am. I'm sharing. Mm, 1 second, ma'am. Actually, my computer is not working.
You can orally tell your title
No,
and
I'm
conclusion.
just sharing. Wait for 2 minutes. I'm just sharing.
Okay, we can call out. Okay. Yes, Rashmi, your PPT is, your screen is visible actually, not your PPT.
Hey, hey, I can present after 5 minutes. After.
Okay, we'll call out next participant, Mr.
Okay.
Gobit Prasad, BBMKU, Dhanbad, Jharkhand. Mr. Gobit Prasad, are you there? Miss Jyoti Yadav, Research Scholar, University of Allahabad.
Yes, ma'am. Govind start.
Okay, Gobit Prasad, please share your PPT.
Okay, ma'am. I am sharing.
Participants are requested to keep your PPT ready.
Ma'am, slide is visible.
No, your PPT is not visible, Gobit.
Ma'am, visible or not?
No, not visible yet. Oh, okay, you can take your time to sort out technical issues. We'll give you time in the last.
Ma'am,
Miss, Next participant, Miss Jyoti Yadav, Research Scholar, University of Allahabad.
Yes, ma'am.
Please share your PPT, Jyoti.
Yes, ma'am. Govind.
अब गोविंद आ गए. Okay, Govind Prasad has started presenting. Please present. Govind Prasad, start your presentation.
Ma'am, I can present. Rashmi Kesarwani.
Govind Prasad, am I audible to you?
Yes, ma'am.
Govind Prasad, your PPT is not visible now. We will give you a chance in the last now. Please hold on. Rashmi Kesharwani, you can start.
Yes, ma'am.
Rashmi Kesharwani, share your PPT if you are ready.
Okay, ma'am. Ma'am, my PPT is visible.
Yes. Please directly come to the conclusions. Tell your title and tell the findings and conclusion of your work. Straight away.
Okay. Good morning, everyone.
Okay.
Good morning, everyone. Myself, Rashmi Kesarwani from the Banaras Hindu University. I am giving my presentation on the topic improvement in hydrogen storage characteristic of magnesium hydride using dual autocatalyst MgF2 and CHS. Outline of my talk is introduction, metal hydride of for hydrogen storage advantage, nanocatalysis, synthesis, characterization of hydrogen absorption, studies of mixed magnesium hydride, and conclusion. As the world need of energy is growing day by day, there is a need of sustainable energy. In with the hydrogen energy and wind, solar, and nuclear is the main power, and we are mainly focusing on the storage part of the area. As we can store hydrogen in the form of the metal hydrides we can store the hydrogen in the form of the gas and the liquid. Metal hydride is the safe and the efficient hydrogen capacity and vice versa. Metal hydride are formed when the hydrogen
Rashmi, your voice is not clear.
My voice is not clear, ma'am.
Yeah, it is cutting in between.
Metal hydride are formed when the metal atom forms a
Please tell the
stable bond with the
conclusions of your work, Rashmi.
Yes, ma'am. Yes, ma'am. Metal hydride are extremely safe and ease of operation. Refueling can easily be done. As an example of the volumetric and hydrogen advantage of using metal hydride is long-term stability, long life, fast refueling, safe storage due to the low pressure system, compact in size and portability and mobility. What is the nanocatalyst? Catalyzing in dissociation of the hydrogen molecule or recombination of the hydrogen atom. Ball milling is a simple and effective way to introduce and disperse the catalyst into hydride and thus they generate the nanocatalysis. Various transition metal oxide, non-metal and compound plays a catalytic role in hydriding and dehydrating reaction of Mg. Activation energy of MgS2 decomposition was found to decrease significantly owing to the MgS2 has a complementary properties as a previous study shows that there is a desorption when the MgS2 has a higher when the MgS2 is mixed with the other fluorides or other then there is an improvement in the absorption characteristics. In our process, we have mixed MgS2 with CSF 7-8% and ball mill for 24 hours and ball mill for 24 hours at 10 atm hydrogen pressure. And these are my results. These are the XRD results of my XRD SEM and
Please tell the
XRD and SEM.
conclusions, Rashmi.
Yeah, the conclusion that we got 106-degree lower temperature when we mix the MgS2 with CSH. And these are the dehydrogenation and rehydrogenation studies. And these are the cycling stabilities. And these are the thermodynamics studies.
Please come to the conclusion slide, Rashmi. Your time is over.
Conclusion is when we mix CSF with, there is an extreme decrease in the temperature, and Mg, there is a decrease in the enthalpy also, 66.61%, which we got. And I'm very thank you to everyone.
Okay, thank you. Next participant, Mr. Govind Prasad, are you ready now?
Yes, ma'am. I am audible.
Yes, you are audible. Try to share your PPT.
Ma'am, PPT are visible?
It is not visible. Okay, you can tell your title and conclusion of your story work like that only. Without PPT
Yes,
only. Please start your presentation.
well, a very good morning to all of you. Myself, Govind Prasad, PhD scholar, BBMTU, Dhanbad, Jharkhand. And very thankful to Rani Durgavati Vishwavidyalaya, Jabalpur, to give me opportunity in first international conference on nature and natural sciences. Their theme is green technology and socio-economic sustainability. Now come to the topic. My title is a study of biological potential of extract from the leaf sample of Acaranthus aspera. And the background study of their research. The research study provides some insight to the new age herbal medicament with the importance of microbial synthesis and modification of plant extract, and the green synthesis of the nanoparticle, wherein they can be used in relation to development of the new pharmaceutical drug compounds that are more effective, safe, and reliable, and no toxicological side effects. Well, based on research design, based on the quantitative analysis and qualitative analysis, in vitro analysis, by the using FTR, IR, HPLC, and green synthesis of silver nanoparticles, characterizations of silver nanoparticles, and also in vitro analysis of different extracts. Well, my result identification of endophytic fungus associated with the root of the Acaranthus aspera was found to be a Cladosporium species, and qualitative screening of secondary metabolites, and also result of quantitative estimations. PPT all are available in the PPT, but not properly showing. And my conclusion is natural derived active compounds have evolved over millions of years and have chemical diversity that result in the varied biological and pharmacological aspects. These resources have become an integral part of the development of new drugs. These compounds and scaffolds, natural products play a pivotal role in the discovery of the drugs for the treating human disease, especially critical disease. Herbal-based drug system has richer sources of data from the clinical observations and clinical experiments, dose and dosage that give insight to the safety and efficacy of the drug system. Herbal drug system has better safety, efficacy, less adverse effects, compatibility, bioavailability, better cultural accessibility, easy material availability. That this drug system a new dimension in the modern world of therapy. Herbal extract and plant also potential for the synthesis of silver nanoparticles. These are more eco-friendly, greener technology, and efficient as compared to other physical and chemical method of synthesis of nanoparticles. Integral of nanotechnology to the therapeutical prospectus, so increasing the potential, thereby concluding that this technology can be better explored for more efficiency and potential. Thank you.
Okay, Govind, thank you. Next participant, Ms. Jyoti Yadav, University of Allahabad.
Yes, ma'am.
Please share your PPT.
Yes, ma'am.
शेयर हुआ फाइनली, इसलिए पहले से बहुत कर देना।
Ma'am, am I visible?
Your screen is visible. Open your PPT.
Yes, ma'am. Just a minute, ma'am.
Yeah, it is visible now.
Okay. A very good afternoon to everyone. Today I am Jyoti Yadav representing my topic gamma-induced cytomixis and its consequences on meiotic and post-meiotic products of pollen mother cells of Quercus capsularis. When I'm talking about the area of concern, my plant, which is Quercus capsularis, is a plant which is having natural fibers, natural bast fibers, which is phloem fibers, and it is used in making some biodegradable products which can directly relate to the removal of plastics from the environment, which is a very big threat for our environment. And these are some data which can clearly shows how carbon sequestration and carbon footprinting via biodegradable natural fibers can replace the toxicity and the hazardous effect of polythene bags from the environment. And the introduction of my plant was, it is a jute Quercus capsularis and belonging to the family Tiliaceae, and its chromosome number is 2n is equal to 14, and its chromosome number is very low or less in number, due to which the genetic variability via conventional method is very less or very low. So we can induce some varieties via induced mutagenesis to have some varieties which are having desirable characters, which is low lignin content product, low lignin content fiber, because jute fibers have high lignin content, due to which its durability is very low for further textile industries and other type of chemicals. As per its economic importance, now currently having in carry bags or in packaging and other some handicraft
Jyoti,
material.
please tell conclusion.
Yes, sir. Yes, okay, ma'am. My main topic is cytomixis, and cytomixis is the phenomenon in which immigration of chromatic material through cytoplasmic channels or intercellular bridges between proximity of closed meiotic cells. And due to this type of due to this method, we can found cells in sites which are having high purity level than normal cells. And here is the results of my my work, consequences of cytomixis, in which we can clearly see in the data that there are different types of abnormalities present via the cytomixis, and each and every stages of the cell cycle, which is metaphase to anaphase, telophase, metaphase to anaphase 2, and telophase. And here some micropictograms which can clearly show how the chromatic material were transferred from one pollen mother cell to another, creating some via direct fusion like in A, B, C, D, or via in a chromatic thread or chromatic channel in E and F, and via multiple cell fusion like in G and H. And these are some micropictograms which clearly show how chromatin materials are transferred from channel formation via one PMC to the other. And in D, you can clearly see all the chromatin material were transferred from the donor cell to the recipient cell, which can further cause in aneuploids and hyperploid cells. And here are some consequences where you can clearly see there is a bridge formation, there are some aneuploids, there are some hyperploids. And in D, you can clearly see these pollen mother cell was completely empty after the transfer of their chromatin material. And after the consequences in pollen mother cell, we can also see the post-meiotic products, which are tetrads. And in tetrads, we can found monad, triad, dyad, pentad, hexad, and some other type of variation. Like in last 4 to 5 plates, we can see there are some small tetrads were present with the small nucleus were present with the tetrad. And in this, you can clearly see how pollen size and how pollen size.
Jyoti, please conclude your work. Time is running out.
Yes, ma'am. Variation in pollen size and morphology of cytomixis. In conclusion, cytomixis is a common yet erratic cytological and genetic, physiological, and informational phenomenon. This is specific to both healthy and diseased vegetative and generative cell. Via inducing aberrant meiosis, hyperploid, hypochromosomes, and microsporocytes, unreduced and aneuploid pollens, which are
possibly serve as a material for selection in both positive and negative, and make up as a tactical reserve for possible evolutionary events. And via this process, if we can found any variation in the chromosomal level, we can create, we can found varieties which are having the desirable characteristic, and due to, and via this we can protect our environment from these kind of unused or non-biodegradable material. And as a opportunity of natural fibers, when we're talking about natural fiber, it is very important and expands. There is a new opportunities for hard fiber and jute to reach high and valuable markets, and the scope of possible uses of future fibers are
Jyoti,
enormous.
your time is over.
Yes, ma'am, and these are references. Thank you.
Okay, thank you.
Yes.
Next participant, Ms. Diksha Tiwari, Nehru Gram Bharati University, Prayagraj. Diksha Tiwari, are you there?
Yes,
Start
ma'am.
your
Yes, ma'am.
presentation.
Yes, ma'am. Ma'am, my PPT is visible.
Yes, your PPT is visible.
Okay, ma'am. Good morning, everyone. My name is Diksha Tiwari from Department of Zoology, Nehru Gram Bharati University. My title of the presentation, Feeding Profile of Indian Major Carps, Labiorhoita and Sirenus mrigala from Balan River in India. Uh, The Indian major carps are the most preferred farm fishes because of their fast growth, market values, and higher acceptability to consumers. Indian major carps are the most cultivable fish species in India, contributing about 87% of total freshwater aquaculture producing of the country. These species has also shared good proportion in Ganga River system. The biology of these Indian major carps, mainly these are herbivorous, specific food preference at different life stages. Eats mainly zooplanktons. As it grows, eats more and more phytoplankton. Juvenile and adult is herbivorous and column and bottom feeder eats mainly phytoplankton and submerged vegetation. And this is the result of my research. These are the some species present in the gut of Labiorhoita. The major food of carps consist of sand, mud, algae, and decaying material. Labiorhoita is a column feeder fish. The adult are herbivorous, but young fry feed on zooplankton. Fingerlings feed vegetable debris and minute plants. Adult feeds vegetable debris and mud. These are the some species present in the gut content of Serinus mrigala. Serinus mrigala is a bottom feeder fish. It is an omnivorous type of fish. Adult feed on algae and some vegetable detritus and debris. Fingerlings feed on vegetable debris, unicellular algae, detritus, mud. They also feed on rotifers, insects, and larvae, uh, uh, their larvae, crustaceans, etc. These are the some findings. And the result obtained during the present study clearly demonstrate that feeding habitats and of the Indian major carp varies from species to species. Labiorhoita is a herbivorous fish feeding on mid-water, on surface area, on algae and plant material.
Diksha, please
Whereas,
conclude your work.
oh, okay, ma'am. The gut content analysis showed that the availability and preference of food by fish helps to find out the feeding habit of the fish and accordingly fisheries management in the water body. On the basis of present study, it can be concluded that the adult Labiorota is herbivorous and column feeder preferring phytoplankton and submerged aquatic plants. It is reported that the Labiorota had a positive selection for most phytoplanktonic organisms as well as submerged macro vegetation. In this present study, decay matter was the obtained food component followed by phytoplankton, zooplankton, plant materials, and insects in the fish gut. On the basis of these observations, it can be concluded that the experimental fish Mrigal Sirenus mrigala is a bottom feeder and omnivorous in nature. These are the some
Okay.
references.
Thank you, Diksha.
Thank you so much, ma'am.
Okay. Now we have an invited talk in between for 10 to 15 minutes. After that, oral presentations will be continued. We have our invited speaker today, Dr. Mrs. Sachdeep Kaur from Texas, USA. Madam, are you there?
Hello, good morning, everyone. Yes, I'm right here. I'll be sharing my screen. Give me a second, please. Okay. Can you see my screen?
Ma'am, your screen is not visible. Yeah, now it is visible, ma'am.
Okay, great. Awesome. Um, so myself is Sachdeep Kaur. I'm an educator at CFIST, Texas, USA. And the topic of our research is wetlands, an ecological boon for the environment. The primary intent of this research is to raise awareness about the importance of wetlands, about the wetland delineation, wetland benefits, mitigation, and wetland biology. Once wetlands were considered to be useless and disease-ridden places, but now we have enough research that proves that they are no longer useless areas, rather they are an ecological boon for the environment. I'm gonna just jot down the importance of wetlands in 3 main points, which is they absorb water, they filter water, they can be called the kidneys of our earth, and they provide habitat to many species. And wetlands are the places that collect waters where the heavy rain and the snow melts and it deposits. They hold water and they avoid flooding. Wetlands are just like the sponge. They soak into the groundwater and they maintain the groundwater levels. Number 2 is they filter water, and they can be considered as the kidneys for our earth. They hold up to 90% of the soil that was eroded and carried by the runoff. Bacteria in the wetland soils can be broken down and it reduces the chemical pollution. And number 3 is the habitat. So wetlands provide a home for a variety of living things like plants, animals, birds, reptiles, insects. The marsh is a wetland that has grasses and different types of plants filled with soil over time. And now I will come to the conclusion. So the conclusion for this was this: in this research, we tried to highlight the incalculable value of wetlands for our environment and the threat that the chemical and the climate change and human interference poses to them. We now realize that many of our wetlands have been destroyed or degraded due to the development, misguided engineering projects, agriculture, and fossil fuel development. We are just beginning to understand the vital services that wetlands provide and are designing the blueprint for restoring these precious gifts of Mother Nature. Thank you, everyone. I think that was all about our conclusion and our presentation.
Thank you so much, ma'am.
Thank you.
We are grateful to you for your presentation. On behalf of International Conference on Nature and Natural Sciences, all the organizing committee, I express my gratitude to you for presenting here.
Thank you so much. It was a great privilege to come and present on this platform, and I will look forward to have more opportunities to come and present my research. Thank you. Have a great day, everyone.
Thank you so much, ma'am. Now moving on with the oral presentations. Next participant, Miss Megha Desai, the Maharaj Sayaji Rao University, Baroda. Megha Desai, are
Hello.
you there?
Yes, ma'am. Can you hear me?
Yeah, please share your PPT.
Yeah. Good morning, everyone. So today I'll be speaking about management and prevention of environmental pollution by proper disposal of unused drugs. So my contents would be introduction, objectives, materials and methodology, work done, outcomes, and references. So basically, the presence of pharmacological active organic micropollutants are emerging issue of great concern at present. Pharmaceutical compounds are basically APIs, which are biologically active ingredients in pharmaceutical drug and specifically designed to interact with biochemical pathways. So these, when they are in the human body, they have to resist the biochemical degradation. So these are called environmentally persistent pharmaceutical pollutants. And when they are released in the environment, then biological activity of environmentally these EPPs may directly adverse effect have on non-target organisms such as wildlife, ecosystem health and resilience. So disposal of unused drugs, pharmaceutical product facilities, hospital waste, human excretions, and veterinary applications are the origin of pharmaceutical pollution. So this is the basically the sources where we can see from the picture that from the sewage or from the manufacturing site or from the farming or from the directly releasing the effluents to the river water bodies, we are reaching the groundwater and the surface water. So effect, there is a great concern of this effects which are having on the target organisms of these APIs and on the non-target organisms, the like fluoxetine, NSAIDs like diclofenac, cytotoxic drugs or chlorpyrifos or atrazine. So, according to the USA FDA guidelines, there are some safe disposal. There are some guidelines for disposing of the unwanted pharmaceuticals in and after emergencies for at a country level. So these are the disposal methods: incineration, landfilling, encapsulation, inertization, and Simetkins. And at the household level, so we have to identify, this is also according to the US FDA guidelines, that whether you have a drug take back system available in your area, nearby area, or do it fall in FDA flush list. If no, then you have to go your medicine dispose of to the drug take back system. If yes, you can directly flush your medicines into the toilet. So my objective was to mainly focuses on identifying the practice and disposal method of commonly unused medicines by household from various parts of Vadodara. So I prepared a questionnaire based survey I
Tell
did.
your conclusions.
Yeah. And like this, it was awareness come educating the people, and it was descriptive research method, and I used Microsoft Excel and SPSS software, and literature survey revealed that disposal of this is is used by incinerators, so we cannot, it's not environment friendly, so they produce toxic gases. So it is better that we can, like I conducted questionnaire of 60 households
Please
where I.
conclude, Megha.
Yeah, ma'am.
Your time
Yeah.
is already running out.
Yeah, ma'am. So during the survey, people were educated to understand the importance of properly disposing of expired medicines, and there are now programs in my city also that that have collection points. They call it drug take back system, where they collect the drugs. Grocery stores are there, police stations are there, where we can where we can give them and they can dispose of according to the guidelines. These are my references, and thank you.
Okay, thank you. Next participant, Ms. Deepika Bamal, Chaudhry Charan Singh, Haryana Agriculture University, Hisar. Ms. Deepika. Next, Ms. Divya Pipalde.
Yes, ma'am. Good morning, ma'am.
Okay, please share your PPT, Divya.
Yes, ma'am. Good morning, दिख रही है, ma'am? Hello.
Hello.
जी, ma'am.
Yes, your PPT is visible. Please directly tell your conclusions, title and conclusion.
जी, ma'am. The topic of my research is special characterization of soil pH, EC, and organic carbon in Jajmao Industrial Zone, Kanpur through interpolation model. Jajmao Industrial is leather and tanning industries, so it is highly contaminated by heavy metals. So we study pH, EC, and organic carbon, which are essential parameters used as primary health status of soil. We use technology, geographic information, and GPS. By this is eco-friendly methods and cover a large area. So details of my work: soil samples collected by grid-wise at the interval of 500 meter, and laboratory analysis of pH, EC, and organic carbon are used with these standard methods. And for soil preparation of fertility maps, we use ArcGIS, in which we study semi-variogram and spatial variability of interpolation model, in which we use three models: Gaussian, exponential, and spherical model. The findings are that semi-variogram models, and the best model is selected based on the following criteria, in which MS should be nearest to zero, root mean scar stability should be lowest, average standard error should be nearest to RMSE should be nearest to 1. So the conclusion of my result is that there are three interpolation models in which we study. A Gaussian model is best for organic carbon, and pH and EC is still a best exponential model. Thank you, everyone. Thank you.
Thank you. Next participant, Ms. Shivani Bhati, the Maharaja Sayaji Rao University, Baroda.
Yes, ma'am.
Please share your PPT, Shivani. Shivani Bhati.
Hello, is it visible, ma'am?
Yes, Shivani.
Yeah,
Please tell
okay.
title and conclusions.
Okay. Good morning, everyone. Myself Shivani Bhati. My title is Effect of Natural Ventilation System on In Vitro Shoot Cultures of Eclipta alba. We studied the effect of air diffusivity in the culture vessels on growth of Eclipta alba plants. Air diffusivity was obtained by various membrane systems with different pore sizes. This allowed the environment inside the culture vessel and outside to be similar. This reduced the problem faced in closed cultures conditions such as abnormal growth, vitrification of cultures, significant death rate during the acclimatization phase. So, in conclusion, these were the results obtained with various membrane systems. So, the natural ventilation system with one hole with two layers of surgical tape is found to be the most efficient setup for obtaining healthy plantlets of Eclipta alba, which has the highest pore sizes. As an advantage of using this system, healthy longer shoot growth, easy acclimatization, low death rate, high chlorophyll content was obtained. Thank you.
Thank you, Shivani. Next participant, Dr. Rani Arora.
Hello, ma'am. I'm here. I'm Dr. Rajni Arora. It's wrongly written, ma'am. I'm just sharing.
Madam. Please share your PPT.
Yeah, I'm sharing. Ma'am, is it visible? Hello? Ma'am, is my slide visible?
Yes.
Okay. I'm Dr. Rajni Arora from Swami Shraddhanand College, University of Delhi, and I'll be talking on coastal wetlands of India, the problems and strategies of management and its conservation. With the main objective is to highlight the loss of our wealthy and productive ecosystem and to look for some effective strategies for its conservation. And these are the contents which I will be sharing here. The wetland, which is considered to be the productive ecosystem, where I will be focusing on the coastal wetlands, and in the coastal wetlands I'll be highlighting on the mangroves and the coral points. And here the 2nd Feb is observed as a World Wetland Day, and according to Ramsar conventions, wetlands are very, very important, and the government and intergovernmental, national and international cooperation is required for its conservation. This is the list of the wetlands which have been recognized under the Ramsar sites, and the 7516 kilometer, and there are 3497 coastal wetlands present here, and it is listed here in this map of India, both the coastal as well as the coral reef, which I will be highlighting. And the highest, here the coastal mangrove point will be discussing, where West Bengal is having highest number of the mangrove plantation.
Madam,
The picture, yes, just coming to the threats to the coastal wetlands were based on the anthropogenic as well as the natural processes, and there are shrinking mangroves, the number of effects of the tsunami observed in different areas. Same way the corals are affected by coral mining, agriculture, pollution, overfishing, blast fishing, and there is coral bleaching where the coral polyps, they are expelled, expelling their zooxanthella. And the conclusion, there is a need for a multi-pronged strategy for the conservation and management of the coastal wetlands. I am skipping all these. And my conclusion is the coastal wetlands are constantly degrading. The eastern coastal wetlands of India are more affected by natural disasters, and we have lost around 73961 hectares over a decade, and research on coastal wetlands in India is very, very limited. There is effective enforcement which is required for regulations, which is lacking, and there is a need to make an effective and comprehensive policy for the conservation through local people participation and legislative policies with scientific knowledge. Artificial planting, conservation measures have to be improved, and more research has to be done and need to check the anthropogenic activity. Therefore, let us stop cruelty to coastal wetlands before it is too late. Check the anthropogenic activities which we can do and save ourselves from their revenge. These are the references, and thank you for patient listening to me and giving me the opportunity to share.
Thank you, ma'am. Next participant, Satya Pandey.
Hello, ma'am. Good morning, ma'am. Good morning, sir. I am Satya Pandey. Today I am here presenting on topic.
Share your PPT, Satya.
Ma'am, I am audible and visible, my PPT?
Yeah, your
Hello.
PPT is visible.
Okay. Thank you so much, ma'am.
Please
Today I am here.
Conclusion.
Yes, ma'am. I am presenting the topic explicating the role of mutation breeding in chia, Salvia hispanica, to foster food security. First of all, we conclude what is the foster food security and how is the very helpful for us to provide the sufficient and nutritious food to meet their dietary needs for active and healthy life. For that, we selected the chia seed, which is commonly, botanical name is the Salvia hispanica, commonly known as chia seed, which is very helpful for our cholesterol and weight loss properties, because it contains the main composition which chia contains is the alpha linoleic acid, is mainly omega-3 fatty acid. Here, because chia seed contains so much properties like minerals, fatty acids, phytochemicals, that's why chia seed is called as a superfood in daily routines, and also called as a pseudo cereals. In here, I am presenting how mutation breeding program is helpful to increase their yield production and increase the new varieties of plant, which is very helpful for us. Through this, we include the UVB treatment, and which is very helpful. These are the different morphological observations which I found in my study: normal, control, xantha, semi-xantha, and different types of abnormalities. We clearly see in H there is a fused axis, which clearly in my work it shows that there is a normal is a single axis, but being lower doses of UVB exposure is increment in their axis, which is very beneficial for us. In the height trend of this is the M2 generation graph, which is I am presenting to you, that how the normal control plant and with their low doses of UVB exposure in increment in the height of plant, and in the second peak you clearly see the bushy plant, which in which there is just so much branching pattern. We clearly show that how so much branching than the normal plant and how they increase their yield at the low doses of gamma UVB exposure, but at the high doses there is a decline in the height of the plant, which is you clearly seen in the plant height trend.
Okay, please tell conclusions now.
Yes, ma'am.
Satya?
Yes, ma'am. Okay, okay, ma'am. These are cytological observation which I am concluding the after fixing the bud of chia seed, and these are the normal metaphase, anaphase, enough, and different types of chromosomal abnormalities which I found that. These are the cytological table, which in clearly shows that the lower doses of at 10 minutes exposure of UVB exposure, there is a low percentage of F, and there is a low slightly decline in the polyfertility as normal control. And how is the low doses of UVB exposure is beneficial for us? After these all the work, I am concluding that at low doses of gamma is very beneficial and increase their height of the plant, seed size, and different parameter which I am found my work. And the cytological aberration which caused by UVB radiation change their genetic makeup of chia seed can be inherited a subsequent generation to create agronomic superior mutants to be promising material for plant breeder in near future. That's why we are concluding that at the low doses of UVB exposure is very beneficial for us, and our farmers also suggested that variety to develop, and my work is based on the mutation breeding program. Thank you so much, ma'am. Thank you for it. Thank you.
Thank you, Satya.
Thank you so much, ma'am. Okay.
Next participant, Dr. Nirmal Chandra Biswal, Associate Professor, GIET University, Odisha. Dr. Nirmal, are you there? Okay. Next participant, Miss Ish Sirjan Kaur Chandok. Are you there?
Yes, ma'am. I'll just share my screen.
Please share your PPT. Ish Sirjan, please share your PPT.
Yes, ma'am. Can you hear me?
Yeah, you are audible.
Thank you. I'll begin. Hello everybody. The topic of my presentation today is revolutionizing natural science research with ChatGPT. For example, for everybody who don't know, ChatGPT is a new AI technology that has come up. It's done by the Open AI industry, and what it does is that it has a UI which it has a website that you can access, and you can put in your chat. You can put in natural language the way we talk. So, for example, a scientist can put in information what he would say to an intern, and ChatGPT does that work. And I know this is a little redundant, but my understanding of natural sciences is that it's the science that revolves around our biology, chemistry, nature, and it helps us to, it has been the most classic science. This is where our Ayurveda, our basic nomads began science from. So it is the very basic root of science that we talk, and it is the place that we should start when we are revolutionizing. And now an AI that can do mundane work like make you a to-do list, or how it's helping the world right now, making them diets or something like that, and how a classic science then such as natural science that has been there from the beginning can be interlinked and that can be moved forward. So for this we need to understand what is fair data. So fair data is findable, accessible, interpretable, and reusable data. What we understand from this is that we get a lot of data from our patients, from the hospitals. There is a lot of proprietary data. There are online sources such as GEO, TGCA, where all of these data is present, and a lot of scientists use that data to to do their research. And that data is only usable if it is, number one, findable. So it is on these sites that the scientists use. Now, number two is that it should be accessible. So it should be.
Srijan, please tell conclusions.
Okay, ma'am.
Conclusions of your work.
Yes, ma'am. So one of the main conclusions of my work is that the data that is used in natural science, such as in the geology research, in the climate research, it can be detectable by humans, and it can be used on ChatGPT to revolutionize the fair data, and it can help in a lot of research and the future possibilities of linking chat GPT and nature science and fair data can help us to first of all decrease the time taken in research. It can help in improve data collection and analysis. It can innovate our research approaches. So ChatGPT has all the information that is present on Google Scholar and Google, so it can help us on how different literature reviews have done this research, and can do that, and it has a lot of possibilities on creating our whole work. So the conclusion at the end would be that you can enhance our data collection, improve our data analysis, and innovate research approaches with ChatGPT. And the benefits of it can be that we will have more research, more literature reviews, which can enhance our whole concept of nature sciences and take us forward a step into this. Thank you.
Thank you, Ish Srijan. Next participant, Shri M.B. Honori, Scientist C, Institute of Forest Biodiversity. Shri M.B. Honori, are you there? Next participant, Narendra Kumar, Chaudhary Charan Singh University, Meerut.
Yes. Can I audible?
Yes, you are audible, Narendra.
Share a PPT.
Please share your PPT.
Can you see here?
Yes, it is visible, Narendra.
In fact, my research topic is impact of human activity on soil health and vegetation along the Kali River bank in Meerut region. Introduction. The soil health and vegetation diversity are essential components of ecosystem that provide various ecological, economical, and sociological benefits. Soil is the fundamental for agriculture, and its health is critical for producing food, fiber, and fuel. Vegetation provides various ecological services such as carbon sequestration, water regulation, and biodiversity conservation. However, human activities such as agriculture, urbanization, industrialization have been identified as significant driver for soil degradation and loss of vegetation diversity. Kali River is important in the Meerut region that is located in Uttar Pradesh, India. The river is vital source for agriculture, industrial, and domestic purpose. However, the river is surrounded across the areas have been subjected to various human activities that led to the degradation of river ecosystem. Human activities such as industrialization, urbanization, and agriculture have been identified as significant driver for soil degradation and vegetation along Meerut material and method results and then discussion. Results in the funding indicate that human activity can cause considerable changes in soil characteristics such as pH, organic matter, nutrient levels affecting vegetative diversity. Nitrogen and phosphorus level were higher in mixed urban and agricultural land uses compared to higher urbanized and rural areas. Further areas that human activity led had to lower vegetation composition and species richness than those with low human activities. The significant alteration in soil properties and vegetation diversity indicate that human activities, particularly urbanization and agriculture, had adverse effect on river ecological well-being. The study underscore that necessary implementation sustainable land use method to reduce the negative impact of human activity on the river ecosystem. Conclusion. The study finding demonstrate the soil pH were notably lower in highly urbanized areas than rural areas. Furthermore, highly urbanized areas and significant lower organic matter content that areas with mixed urban and ecological agricultural land uses in rural areas. A study has also discovered that nitrogen and phosphorus level are considerable higher in areas with mixed urban and agricultural land uses than in highly urbanized and rural areas. Additionally, veg- vegetation composition and species richness are lower in highly urbanized area compared to areas that mix urban and agricultural land uses in low rural areas. The research emphasized the importance of developing effective management and conservation strategies that can balance human activities, economic and social benefit with the need to safeguard and conserve valuable natural resources. This is the reference. Thank you.
Thank you. Next participant, Soni Chauhan, Research Scholar, CSIR, National Botanic Institute. Soni Chauhan, are you there?
Yes, ma'am. Am I audible?
Yeah, please share your PPT.
Okay, ma'am. Am I visible, ma'am?
No, your PPT is not visible and the voice is also echoing. I think through two connections you are joining. Yeah, unmute your microphone. Your PPT is not visible still. Soni, can you hear me?
Yes, ma'am. I'm hearing you.
Try to share your PPT.
Now?
Yes, now it is visible. Okay. Please start and concentrate on conclusions.
Okay, ma'am. So my topic is integrated application of trichoderma and carbendazim effects, the carbendazim extractability and microbial functions in the maize rhizosphere. And recently I published this paper in Journal of Soil Science and Plant Research journal. So my topic is basically based on pesticide pollution, which affect the microbial diversity in soil and other soil metabolites. So carbendazim, this is a systemic pesticide, benzimidazole fungicide, a metabolite of bionimal, and it is carcinogenic for humans also. So in which we have to experiment that I have some sample of fallow soil and maize cultivated and microbial inoculum, and in different treatments I have checked the microbial diversity in the presence of carbendazim. एंड दिस इज अ ग्रीन नेट हाउस कंडीशन, जिसमें हमने बायोलॉग किया और HPLC करके देखा कि बायोलॉग जो शॉर्ट और शूट रूट लेंथ लिया और एस्टिमेशन ऑफ कार्बेंडाजिम किया यूजिंग HPLC सबस्ट्रेट यूटिलाइजेशन पैटर्न एंड स्टैटिकल एनालिसिस. So these are the results of culture microbial populations. Structure is carbendazim treated and non-treated soil from maize rhizosphere and fallow treatments was assessed by evaluating the microbial count in different media. The concentration of 0, 10 and 100 mg per kg in soil in different treatment: initial soil, fallow soil, maize rhizosphere soil, maize treatment soil. And this is the biolog study to discriminate among 31. These are the carbon substrate which are preferentially utilized or not utilized by soil microbiota. जो यूज किए गए हैं वो और जो नहीं यूज किए गए हैं वो कम हाईलाइट है, जो ज्यादा यूज किए गए हैं वो डार्क हाईलाइटेड है। ठीक है, तो द राइजोस्फीयर soil as compared to the fallow soil showed a significant increase of diversity indices and evenness, shows the average color development by the average temperatures over an incubation periods. It was noted that AWCD of fallow soil was distinctly lower than of rhizospheric soil. Carbon substrate examined by biolog eco plate method. Okay. And a comparison of the initial, these are the carbendazim accumulation in soil and plant tissue. It is a carbendazim standard and standardized by HPLC. Through the HPLC, the peak, this is the peak C908, it is a carbendazim, and this peak is a degraded product of carbendazim. As a standard, black 1 microgram per ml and gray is 10 microgram per ml standard.
Please conclude your work, Sonu.
Yes, ma'am. Yes, ma'am.
Time has already run out.
Okay, ma'am. And this is the extractability of carbendazim from soil. In this is initial soil, fallow soil, maize rhizosphere, maize trichoderma. In these, we conclude that maize rhizosphere and maize trichoderma treatment, the degraded product and carbendazim found in accumulation and not in bioavailable form, जिससे कि माइक्रोबियल एंजाइम्स जो माइक्रोबियल डाइवर्सिटी है वो उसे यूज नहीं कर पाते, उस पर इस पर कोई फर्क नहीं पड़ता है कंपेयर टू इनिशियल सॉइल एंड फेलो सॉइल. And extractability of carbendazim from plant is में maize shoot, maize plus trichoderma, maize root and maize trichoderma shoot. इसमें we show that the degradation and carbendazim, this is a maintain equilibrium in this treatment. It showed the maize and trichoderma is a efficient treatment for maintain the microbial diversity and soil metabolites and soil fertility. And these are the summaries. The present study conclude the fallow condition prolong the presence of residual pesticide in soil and affect the soil microbial community, whereas biofungicides such as trichoderma facilitates degradation. The present study supports the hypothesis of strategic fallow to reduce the effect of residual pesticide and improve soil health in intensively cultivated field having pesticide contamination. It is proposed that a short-term crop may facilitate carbendazim degradation from soil and reduce its accumulation and deleterious effect of microbes. And in the HPLC analysis of soil and maize tissues reveal increased persistence of carbendazim. Its degradation product in fallow treatment compared to maize with or without trichoderma. The application of trichoderma enhanced the carbendazim removal and elevated the effects of the microbial population. Thus, agriculture important microbes along with short-term intervening crops may provide modest sustainable solution to pesticide and contaminations. And this is my research article published in Journal of Soil Science and Plant Nutrition 2000.
Okay, Soni. Thank you so much. Next participant, Dr. Arun Baburao Savarkar, Associate Professor, BP Arts, SMA Science and KKC Commerce College, Chalisgaon, Jalgaon. Dr. Arun Baburao, are you there? Dr. Arun Savarkar, can you hear me? Next participant, Dr. Manka Mishra.
Yes, ma'am.
NES College. Madam, please share your PPT.
Yes, ma'am. Now it is visible?
No, it is not visible. Your screen is, yes, now it is visible, plus, but please maximize it.
Okay, ma'am. Okay, ma'am. I'm Dr. Manka Mishra. A very good afternoon to all. I would like to thank giving me for such a nice opportunity to present my paper. And my topic is study of physicochemical parameters of fish diversity in Sangram Sagar Pond at Jabalpur. The present study was undertaken to assess the water quality and its effect on fish fauna of Sangram Sagar Pond. Is a large landing water body and covering around 16 hectares of area. The pond is struggling for its extents
due to heavy anthropogenic disturbance and its side effects. The pond is running domestic discharge into its water, and during the present study, 5 sampling station sites were chosen for physicochemical parameters as well as biota for Sangram Sagar. And all the 5 stations were chosen on the basis of different anthropogenic pressure at these sites and also allowed assessment of impact of variable factors on fish diversity. Next one is this is the material and methods, and these are photographs founded in Sangram Sagar Pond. And next one is resultant discussion. The diversity of fishes mainly depends upon the biotic and abiotic factors, type of ecosystem, age of the water body. And the next one is these are the results. And the higher value shows highest is red color, and the lowest value shows yellow color. And these are graphs, and our graph shows the water temperature during 2015-16. The water temperature found it to be good zone, and maximum water temperature was recorded during summer months, and highest is stations S4 during the June season. And the next one is the pH value. The above graph shows the water pH value of water at 5 sampling stations during the year of 2015-16, and same trend of pH was observed. This is the all graphs during my study periods. And conclusion, species diversity is different sampling site indicated that altered habitat and high altitude and supported less fish species, while variety habitat like shallow pool and deep pools are primary primary and the contributing to maximum diversity. Subfamily Cyprinidae emerged as the most dominant group. Therefore, protection of these particular habits is recommended for the conservation and management for the pond and diversity. While the most of fishes were present round the year, some species were found absent during the summer pre-monsoon season, and highest number of fishes were caught during the monsoon season. The larger fishes like labio and tor were found during the post-monsoon season. The present study shows that the historical pond Sangram Sagar is facing serious issue due to anthropogenic disturbance and pollution as well as lack of maintenance by the civil authorities. The physicochemical parameters shows that the water quality is low and getting worse. If serious steps are not taken to improve the water quality, this icon to be Jabalpur city will lose the important freshwater body that is Sangram Sagar. And these are my references. Thank you.
Thank you so much, ma'am. Next participant, G.S. Harrington Deva, St. Joseph University,
Yes,
Bengaluru.
ma'am. Yes.
Please share your PPT.
Yeah, ma'am. Um, okay.
Please concentrate on your conclusions.
Okay, ma'am. Um, um, okay. Good morning. I'm G.S. Harrington Deva from St. Joseph University. I'm going to present a topic on exploring terrestrial and wetland avian diversity in a peri-urban ecosystem of Chelegata. Um, I'll give you an intro to Chelegata. It's a village located in the southern part of Bengaluru. It's a multi-divergent ecosystem which has terrestrial, aquatic, pond, agriculture, sewage, etc. Even though the place has a good biodiversity, in recent days it is subjected to intense industrialization. Some spot of the place is experiencing severe pollution in the form of solid waste dumping by the local residents. Nearby NH48 highway roads pose a threat by noise and light pollution. This is the overview of the place. And study area was, as mentioned earlier, it is in Chelegata. The duration was it's not planned study, it's an opportunistic survey from 2022 to 2023. Morning hours were 6 a.m. to 10 a.m. and evening hours were 5 p.m. to 8 p.m. The methodologies used were visual and auditory observation, binoculars and DXLR camera were used to document that. For identification, we used Birds of Indian Subcontinent 2011 edition and Merlin's Bird ID app. This is a map of satellite view of Chelegata, IMAP view of Chelegata, and a view that shows the water bodies of Chelegata. Coming to results and discussion, we observed 75 bird species, which is quite a great number, belonging to 44 families. And the given families are given in text and a visual representation. Here you can see a pie diagram of the graphical diagram of graphical representation. And the next diagram shows the feeding build of the birds, piscivorous, insectivorous, carnivorous, etc. On the third diagram combines the first two slides and tells in what all habitat the amount of food build were observed. The y-axis is habitat and the x-axis is the food build. Results and discussion. We found piscivorous birds are most frequently observed in the pond ecosystem and certain parts of sewage ecosystem. Insectivorous and frugivorous birds were most abundant in terrestrial ecosystem. Granivorous and nectivorous birds were most common in agriculture. Scavengers were observed in wetland. And omnivorous and ophiophagus birds were found in nearly every ecosystem we observed. And despite the area being in biodiversity, intense urbanization in recent times, whereas metro construction and few other urbanization has posed a threat to this bird population. We even observed heavy metal contamination in the place. The wetland ecosystem filled with plastic, other pollutants also pose a threat to the birds. Additionally, the proximity of NH48, which lies within 1 kilometer range, emitting noise and light pollution, pose a threat to the nocturnal birds in the area. And this is a table what all the birds I found in Chelegata. And this is the birds which I found based on their food guild and their habitats. Um, Here are some photographs of the birds which were observed. And any queries?
No, sir. Thank you.
Yeah, thank you,
Next,
ma'am.
next participant, Miss Pooja Tripathi, Assistant Professor, NES Science and Commerce College, Jabalpur. Pooja Tripathi, are you there? Okay. Miss Disha Chahal, Indira Gandhi National Open University.
Yes, ma'am. Ma'am,
Yes,
Pooja
ma'am. I'm
Tripathi
here.
is, I'm here.
Okay, okay. Pooja Tripathi, madam, are you joined?
Yes, ma'am. I have joined. Please
Yeah.
give me a
Please
minute
share
to
your
share my.
PPT now.
Yes, ma'am. Am I audible and visible, ma'am?
Yes,
Ma'am,
you
am
are
I
audible
audible and
and
visible?
visible, but your PPT you have to maximize it.
Okay.
That's it. Please start,
Is it
madam.
fine?
Madam, please start your presentation and please tell conclusions, title and conclusion of your work.
Okay, ma'am. Okay, ma'am. So, first of all, good afternoon, everyone, and thank you so much for giving me this opportunity to share my thoughts on it. So, here my topic is effect of chemical fertilizer on environment and ecosystem. So, as you have asked me to go on the conclusion, so so far, whatever I have studied and whatever I have got the knowledge about fertilizers and have concluded it, it's like fertilizer are definitely something which enhances the soil fertility and it added a nutrition into it, but surely they harm the soil conditions, its texture, its moisture level, then it creates the air pollution, then it harms the water and eventually the human beings because it definitely goes into the intestine and kidneys to their bile through the water. So, with the conclusion, as what I think is, no, fertilizers are important, surely, but there should be some parameters on which fertilizers have to be studied, and soil levels have to be studied so that at least we should know what soil needs. There should be some things, some soil analysis should be there so that we can got to know that what kind of fertilizer has to be given to the soil, what kind of level of potency, maybe nitrogen, phosphorus, or phosphate needed. So that is what I need.
Madam, have you
Ma'am,
finished?
anything else you would like to ask me?
No, ma'am. No, ma'am. Thank you, ma'am.
Yeah,
Next
thank you so
participant.
much, and thank you so much for listening me.
Okay, ma'am. Next participant, Miss Trisha Chahal, Indira
Yes,
Gandhi
ma'am.
National Open University.
Yes, ma'am. I'm presenting now.
Yeah, please.
Ma'am, is my presentation visible?
Yes, it is visible. Please
Ma'am, my
give your
topic is
title and
examining.
conclusion.
Yes, ma'am. Ma'am, my topic is examining change in the citrus food production in Haryana. The main objectives are to analyze the total area and total production of citrus fruits in different districts of Haryana and to examine the change in the total area and production of citrus fruits in Haryana. The conclusion from the study is that even though the change has happened, but the change is not consistent in all the districts of Haryana. Certain districts show less change in the cultivation of citrus fruits as compared to the others. Districts showing less growth need more incentives to promote the growth of citrus fruits, and the research can be used to promote the crop diversification and the growth in the production of fruits and vegetables in Haryana. And it can also be used by the government to formulate the necessary policy measures related to citrus fruits in Haryana. Thank you, ma'am.
Trisha, thank you. Now I will call out participants who were left. We will give 2 minutes to them. Miss Ankita Rani, are you there?
Yes,
St.
ma'am.
Joseph University.
Yes, ma'am.
Please present Ankita.
Yes, ma'am. Is it visible, ma'am?
No, your PPT is not visible, Ankita. Please try, or you can just tell your title and conclusions orally without PPT.
Good afternoon to all the respected dignitaries, presenters and attendees. I am Ankita and today I am going to present a study on the topic of a preliminary survey to document coordinates along the Sri Krishna Lake, Bayatrandodi, Bangalore, Karnataka. The authors are me and Abhishek Mishra under the guidance of Dr. M. Jayashankar, Department of Zoology, School of Life Sciences, St. Joseph University, Bangalore. Starting with the introduction, dragonflies, also the anisopterans, belongs to the order Odonate. They are the bioindicators depicting the health of ecosystem. They are found in the wide range of habitat of water bodies, and it is an ongoing study to reveal the diversity of dragonflies in the Sri Krishna Lake, Bayathandodi, Banarghatta, Bangalore, India. The materials and methods that we have used, the area of observation was the Sri Krishna Lake, and the study duration, it's an ongoing study started from the month of April 2023. The gadgets that we have used are the DSLR camera, Samsung Galaxy M31s, Realme C15, and macro lens at 12x to 24x.
Please tell conclusions, findings of your result.
Yes, ma'am. We have came across a total of 10 species of dragonfly, out of which 8 were belonging to the Libellulae family and Aeshnoidae and Gommifidae. So as
It is a larger family, so we have recorded 8 species from that, and each one under Rachnoidae and Gomophidae. Here are pictures that we have captured during our observations. And since they both looks very similar, Crocothymus cervilia and Ethymantha brevipenis. The difference here is that the red dorsal black line is present here, and which is not present in the brevipenis species.
Ankita, please tell
Varieties
your
variegata and
conclusions.
Neurothermal stulia.
Please come to the conclusion slide.
Yes. So the results were that dragonflies are found mostly near the water bodies, and total 10 species of Anisoptera were observed, and 80% of were belonging to the Legulidae family, and 2 species were found to be abundant, and the maximum activities were observed during the midday. Here's the pie chart showing the distribution of families. Whereas the discussions are that it is subjected to pollution, the area, so we can study on the basis of pollution, the odonate diversity. And since it's an ongoing study, we can continue it in different seasons to understand seasonal diversity and distribution. And this is the first time observation that has been undertaken. So further studies on endemism and conservation, if there are threatened species, needs to be undertaken. The acknowledgement authors are thankful to Dr. K. Subramaniam, Zoological Survey of India, Chennai, India, for confirming the identification of anisopterans. Here are the references. Thank you for giving me this opportunity.
Thank you, Ankita. Next, Miss Vasundhara Singh. Miss Vasundhara Singh, are you there? Miss Deepika Bamal. Dr. Nirmal Chandra Biswal. Shri M.B. Honori. Okay then, Bhagyashri Biswal, are you there? Bhagyashri Biswal. Bhagyashri, are you there? You want to present?
Yes, ma'am. Yes, ma'am. Just
please make it fast and share your PPT. Start your presentation, Bhagyashri. It is not visible. You can tell your topic and conclusion orally.
Hello. Okay, ma'am. I am Bhagyashri Biswal, GIT University, Gunpur, Odisha, Department of Life Science. My topic is surface modification of Muga silk by
Your voice is not clear, Bhagyashri. Are there any network issues? Hello? Your voice is not clear. Bhagyashri, please start.
Hello?
Yes, you are audible.
Hello, ma'am.
Yes.
Yes. Okay. Ma'am, my topic is surface modification of grass silk by using Sapindus lorifolia. Actually, Sapindus lorifolia is a plant which is used as a degumming agent. That means the Sapindus plant I am using as a surfactant. My topic is regarding silk, ma'am. Textile is a vast field ever growing in the improvement of the field of science and technology, and silk is the queen of textile, is a splendid gift of nature. So silk is a natural protein fiber. In the protein fiber is fibrin and sericin, and here the silk property is that the
color, luster, smooth feeling, toughness and lightweight, innate and affinity of dyes and fitness, strength, elasticity, high observance, resilience and flexibility. These are major and my is in for filament 3 kg so 500 to
Bhageshri, your voice is cutting.
durable.
We are not able to understand.
Hello.
Bhageshri, there are some network issues, I think, at your end. We are not able to understand. Your voice is interrupting in between. Please check your network issues. In the meantime, we are calling next
Hello.
participant. Please check your network issues.
Okay. Okay, ma'am.
Dr. Arun Savarkar.
Okay, ठीक.
Dr. Arun Savarkar, are you there? Dr. Arun Savarkar. number. Dr. Arun Savarkar, can you hear me? Your PPT is not visible and you are not audible. Dr. Arun Savarkar. Next, Miss Preeti Tarke. Arun Savarkar, you are not audible. Your PPT is visible now. Please respond. Dr. Arun Savarkar, can you hear me? Next participant, Miss Preeti Tarke.
Yes, ma'am. Am I audible to you?
Yes, you are audible. Please share your PPT.
Yes, ma'am. Yes, ma'am.
Dr. Arun, your voice is not coming. Please stop sharing and solve the technical issues at your end. Miss Preeti Tarke, start your presentation.
Yes, ma'am. I'm sharing. 1 second.
Miss Preeti Tarke.
Yes, ma'am. 1 second. 1 second. Ma'am, can I say because my
Yeah.
network?
Okay.
My
Please
one.
tell your title and conclusion. Preeti Tarke, are you there? Okay, I think she's facing some technical issues at her end. So, Dr. Arun Savarkar.
Ma'am, hello, ma'am. Am I audible, ma'am? Hello.
Yes, yes.
Am I audible,
Ma'am,
ma'am?
time is over now for the oral presentations.
Ma'am,
I
good
called
afternoon,
out
ma'am.
your name at that
Ma'am,
time.
Ma'am, I'm Vasundhara Singh. I have some technical issues in network. Can I present now?
Don't present, just tell your title and your conclusion.
Sure, ma'am.
2 minutes we are giving because already the time is over.
Okay. Okay, ma'am. Ma'am, my presentation topic is fungal diversity in rhizospheric soil for Osminum sanctum and Mentha piperita. Vasundhara Singh, Department of Botany, Chaudhary and CMP Degree College. Ma'am, mycorrhiza is an obligatory symbiotic association between soil-borne fungi with roots of higher plants. And in my research findings, I am here finding that the dominant active ectomycorrhizal fungi spore species, which is having glomus glomeratum and gigaspora and many other type of species, in which in comparison to both the plants, Ocimum sanctum and Mentha piperita, Ocimum sanctum is more diversity, founding more diversity in comparison to the Mentha piperita. In total, 28 active mycorrhizal fungi species were identified and belong to the 5 different genera. And in all of these, the most dominant fungi was Glomus glomeratum, Gigaspora species 2, Glomus fasciculatum, Glomus aggregatum, and Ecolospora. And mycorrhizal status in the term of both percentage root bit infection and AMF spore population was recorded more in Ocimum as compared to Mentha for all the stages of plant growth. And the highest AM fungi association was recorded during fruiting stage and the lowest of the seedling stage for both the plants in all the selected survey sites. And there was a positive correlation between the AM spores population and the percentage of root bit infection, as active mycorrhizal fungi association is very significant for the production of the alkaloids present in the Ocimum sanctum and Mentha plants. It is a very positive result for this purpose. Thank you, ma'am.
Okay, Vasundhara,
Ma'am,
thank you.
please, I can explain
Yeah.
in 1 minute.
Preeti Turkey, please
Yes,
you
ma'am.
also tell your title and conclusions.
Ma'am, my topic, good afternoon to all. Myself, Preeti Tirki, and I'm a research scholar from Dr. Hari Singh Gaur University. And my topic is protective role of thymoquinone against malathion-induced toxicity in freshwater fish. I have taken malathion toxicity. It is a man-made phosphate insecticide that is commonly used to control mosquitoes and a variety of insects and attack fruits and vegetables. It comes in pure form, color liquid, and other in its technical grade. Malathion is brownish yellow color and it smells like garlic. And my antidote is thymoquinone. Thymoquinone is a bioactive compound of Nigella sativa. It is an annual flowering plant. It is also known as kala jela or karanji. My methods and methodology is I have taken 4 groups. One is control, second is treatment pesticide, third group is malathion along with thymoquinone, and fourth group is thymoquinone. My technique, I have done RAPD PCR. RAPD PCR, I have used 5 primers according to it, and primer OPA and primer OP8 are the shows polymorphism information compared to other primer markers. My conclusion is that RNA polymorphic DNA fragments from PCR amplification. It involves single arbitrary primer in PCR reaction in the amplification of many discrete DNA products. By this technique, we can study the gene variations in different groups of treated and controlled in gene level. This technique is no prior knowledge to the genome and it's need only small amount of DNA. These are my references and thank you, ma'am. Thank you so much.
Thank you,
Hello,
Preeti.
ma'am. Ma'am, I'm Bhagyashree. Can I present? Hello.
Time is over, Bhagyashree. We are already.
Actually,
Please tell
ma'am,
your
मैं तो
title. Yeah, title
my
and key conclusion. Your
okay,
name
ma'am.
is
Okay.
also not there in the list. I think you have not contacted before to Dr. Saurav Gupta.
Yes, ma'am. Just I'm telling my topic.
Okay, please tell.
My topic is surface modification of muga silk by using Sapindus lorifolia as a degumming agent. That means Sapindus lorifolia I am using as an surfactant. And here my topic is regarding silk and silk degumming. Here, what is degumming? Here I am using Muga silkworm cocoon for my experiment. Muga cocoon are formed with continuous filament of 350 to 550 meters production of 1 kg of muga silk, raw silk, 4,500 to 5,500 cocoons are required. The silk is known for its extreme durability and a natural yellowish golden tint, glossy structure. And here, the cocoon is a component of Chinese medicine treated diabetes related diseases. There are other some uses of cocoons. And here it have multiple biological activities and sericin reduce serum lead content improve glucose tolerance. Then modern biological researches and applied in food related products. Then my topic is regarding degumming. What is degumming? Degumming is the process of removing of sericin, a sticky
Please tell
substance produced.
conclusions, Bhagyashree.
Okay,
Conclusion of your work.
yes, ma'am. And my objective is the isolation of different active agent from the chemicals and metal and experimental method. Then my conclusion is that from the above experiment, it was concluded that by using Sapindus lorifolia as surfactant, the degumming loss in sample 1 is 8 millimolar and sample 2 is 8.2 millimolar. The smooth surface of the degumming silk fiber is equivalent to that the other degumming chemicals that are previously described. Thank you.
Thank you, Bhagyashree. With this, we have come to the end of the second day of oral online presentations of First International Conference on Nature and Natural Sciences. After this session, there will be valedictory session, which will be on offline mode from 2 to 4 p.m. at Vigyan Bhavan, RDVV, Jabalpur. I am grateful to the jury members of today's session, respected Dr. Anju Pathak Madam and respected Dr. S.K. Dubey Sir. I am also thankful to all the participants for patient hearing and joining the session. Thank you everyone.